Hello, my name is Michael Hyland, and I'm an international student studying biochemistry at the University of Chester. In this video, I will be taking you through the steps of a cell signaling pathway that regulates countless cellular processes and has shown great promise in the realm of cancer management and treatment. This is the MAPk signaling pathway. Mitogen-activated protein kinase, or MAPk pathways, are an umbrella term used to describe many different cell signaling pathways, much like rock and roll is a term to describe a large genre of music. For this video, we will be using the extracellular signal-regulated kinase, or ERK, as our specific example of a MAPk pathway. Now, all MAPk pathways follow the same general progression, known as a kinase cascade. Before we continue, it is important to understand what a kinase is. A kinase is an enzyme that phosphorylates another protein. For the scope of this video, phosphorylation is simply going to mean to activate or turn on another protein. So, in other words, a kinase is simply a protein that turns on or activates another protein. A kinase cascade starts with some kind of input or stimulus from outside of the cell. In our example of the ERK pathway, this is a growth factor ligand, or a GF signal. This signal is transferred across the cell membrane via a transmembrane receptor. This receptor leads to the activation of the MAP kinase, or MAP3K for short. MAP3K then goes on to phosphorylate MAP2K, which in turn activates MAP kinase. Now, the activated MAP kinase can then go on to perform many different functions throughout the cell, playing a key role in cell growth, differentiation, and proliferation. To put this information into context, I have created an animation that follows the ERK pathway through the cell. Here we have a diagram of the cell membrane, with the extracellular fluid on the outside, the cytoplasm on the inside, and a receptor tyrosine kinase as our example of the transmembrane receptor. First, a growth factor binds to the RTK receptor. This causes a structural change and the recruitment of multiple proteins to the intercellular domain of the receptor. But for the scope of this video, it is only important to remember that this signal causes the activation of the membrane-bound Ras protein. Ras is a crucial protein which instigates the activation of the ERK pathway. Ras is responsible for phosphorylating RAF, which is the MAP kinase, the first enzyme in the kinase cascade. RAF then goes on to phosphorylate MEK, which is the MAP kinase. MEK then activates ERK, which is the MAP kinase. This activated kinase has many functions, affecting proteins both in the cytoplasm and in the nucleus. Let's follow the ERK protein as it approaches the nucleus. Although ERK affects countless proteins in the cell, for this example, let's watch as the activated ERK protein binds and activates two transcription factors, Phos and Jun. After activation, these factors will enter the nucleus and combine in order to bind to a specific region of the DNA code known as AP1, allowing for this string of DNA code to be translated. This process mediates crucial cell functions like growth and proliferation. Now we understand how the MAPk pathways can take signals from outside of the cell and cause all sorts of physiological changes inside of the cell. But why is this important? As discussed, MAPk pathways mediate cell growth, proliferation, and even apoptosis. But when unregulated, these are all factors that can lead to tumor formation. If we look back at the ERK pathway, a common cancer-causing mutation occurs in the Ras protein, causing it to be constantly activated. This means the pathway no longer needs a signal, but can promote downstream effects uncontrollably. As you see here, mutated Ras continues to phosphorylate RAF without receiving any signal from the cell. The continued signal promotes unregulated cell growth and division, which leads to further mutations and eventually tumor formation. Their direct correlation to oncogenesis is why MAPk pathways have become such a promising target for cancer treatments. Hopefully, by developing a better understanding of MAPk pathways and their oncogenic mutations, we will continue to develop even more targeted cancer therapies that have a higher efficacy, less recovery time, and a lower instance of recurrence. Until next time, thank you for watching and stay safe.